Okay, hello and welcome to the HCA BioNet March 2019 seminar on machine learning approaches used in microbiome and metagenomic analysis. A couple of house rules. Please keep your microphones on mute to prevent any static and feedback, such as the one we're hearing right now. The presenter will answer questions at the end of the seminar. These can be typed in the chat box, and Pablo will facilitate the question and answer session at the end of the talk. So without further ado, I have the pleasure of introducing our invited speaker for today, Dr. Chris Fields, who has recently been appointed as the Director for the High Performance Computing at the University of Illinois at Urbana-Champaign. Chris has from the onset been one of the most active pioneer developers for the Biopol project and other notable open source bioinformatics projects that build infrastructure for bioinformatics and genomics, such as the GBrowse and the GBoard projects. Chris collaborates extensively with different numerous open source bioinformatics projects under the foundation of the Open Bioinformatics Foundation, where he is the secretary of the board. As his current position in a core bioinformatics and genomics facility at the University of Illinois, Urbana-Champaign, Chris is at the forefront of developing infrastructure and tools for the high-throughput genomics research and is involved in a number of ongoing microbiome and metagenomic analysis. Chris's talk today ties in with the advance of genomics data generation with the particular focus on the application of machine learning approaches within the analysis of microbiome and metagenomics data. So thank you, Chris, and over to you.
Thanks, Samir. So I guess everyone can hear me okay. Now, our group at the University of Illinois has been working on microbiome and metagenome analyses increasingly over the last couple of years. And the projects that we're seeing have been coming in have been more and more complex, have added more and more samples. The data sets have been more complicated. And so we're invested in trying to find the best way of analyzing this data and getting it ready for researchers in a manner that they can actually make sense of. And some of the best ways to do this are using machine learning approaches. So this is going to essentially be a review talk. It's going to discuss methods that are used, machine learning methods that are used in both processing and analysis of data. It's not going to go into the methods in a lot of detail because there are a lot of different steps in the procedures, and we're going to talk both about targeted and whole metagenome analysis, but it kind of gives you an overview of the applications of machine learning that are used in microbiome and metagenome analysis. So overview is basically what is the microbiome. I'll talk a little bit about targeted versus whole metagenome analysis, and through that I will discuss how machine learning approaches have been used in both applications for targeted amplicon analysis such as 16S and for whole metagenome data analysis. Then towards the end of the talk I'll discuss how machine learning has been used for phenotype prediction, looking at applications where you're trying to assess whether or not a particular sample may be, as an example, it may predict whether a person has a disease state. And then at the very end, I'll discuss possible upcoming challenges in the field with regards to changes in technology and how machine learning may actually be useful for those particular applications. So what is the microbiome? The microbiome is essentially the combined genomic material from microbiota, the organisms that are present from a community, ecological community. This could be from environment samples, so soil or water. It could be from medical samples, oral, fecal, kidney, stone. We recently did a kidney stone project, and yes, there are some things there. You would think it wouldn't be, but there are. It's quite a bit. Industrial, we've seen samples that have come from bioreactors. Whoop, something happened there. Go back. But pretty much anything. As an example, I'll see if this pointer will work. There's this windshield splatter analysis That was done a number of years ago. The Galaxy Project leads wanted to drive around the east coast of the United States and then just gather the gook, the smushed mess that's on windshields, and then literally just sequence it just to see what was there. This kind of gets into what the difference is between the microbiome and full metagenome analysis. So when we are discussing the microbiome, we are really focusing on things like bacteria, archaea, viruses, and then eukaryotic microbes such as fungi and protists. Metagenomics is the study of genetic material from environmental samples, which can encompass more than microbes. It could look at larger eukaryotic organisms. So the windshield splatter experiment actually picks up insects and lots of other things as you might expect. Lots of different microbiome projects, the Human Microbiome Project, HMP. There's the microbiology of the built environment, which is out of UC Davis. I believe this is the one that's headed by Jonathan Eisen's group. Metasub, which is the study of the microbiome in urban environments. Let's see if I can, there we go. See if I can switch that over. And then you have the Shed Aquarium, which is right up the road from us in Chicago, Illinois. And they have their own personalized microbiome project that's looking at samples from the various aquatic environments that are within the aquarium itself. So there's a wide variety of ways that you can study this. But how do you measure complex communities? There are different approaches. There's the targeted approach where we're looking at specific marker genes and the abundance of those marker genes in samples. There is the just basically shotgun sequencing of all of the data that's present. And I'll go over both approaches. But really this is all going towards what the biological questions are. In a broad manner, the biological questions are who is there? Which is the taxonomic diversity in the samples that are present. So basically you're looking for what organisms are there. You're actually just focusing on the diversity and the composition of what's present in the samples. What are they doing, such as the functional information that's present in the samples? So you're actually looking at the genic content. This could be, if you're looking at a metagenome, you're looking specifically at the metabolic potential. And then how are they doing it? This is going into gene expression, looking at whether or not particular genes are being expressed, so whether or not particular protein products are being produced. And it can even delve into metabolomic analysis, so basically looking at what metabolites are being produced. So two general approaches, as I mentioned, targeted approaches. We're going to look at specific markers or a set of specific markers, depending on how you want to analyze the data. And you're going to use that to identify, quantify the microbes that are present, or you're going to sequence everything that's there. We will talk about other meta-omics as well. So, I'll talk about those in relation to how they can actually help inform the metagenome. So we'll start our little tour of metagenomics here next with targeted analysis. So targeted is going to be focusing on estimating taxonomic diversity in samples. We're going to amplify a specific targeted region within a gene. We're going to amplify the DNA in those regions. From that big bag of amplified DNA, we're going to sequence it and then group the reads. So these are going to be grouped in some manner. Those groups are going to be quantified. Then you're going to assign within those groups a rank along with the abundance from the quantification. And then those are going to go into downstream analyses where you can actually make sense of the data depending on what your experimental design is. So this goes into specifically what markers are being used and why. And the most common marker by far that's been used for microbiome analysis has been 16S. Not the only one, but it is the most common one. So why sequence ribosomal RNA? So I'd be completely remiss to not mention Carl Woese. So Carl Woese pioneered the use of 16S to determine the phylogenetic relationships between microorganisms. This is a gene that's present in all bacteria and archaea. It's required. And so this is also, because it has a secondary structure that also has these loops in the secondary structure, it has both conserved and variable regions that are required to retain the structure, but also give rise to a lot of the variability within organisms. So you can design regions or design primers to amplify specific regions of interest. And those variable regions allow you to differentiate between the species that are present. So in a nutshell, you can amplify a specific region. Let's say we want to look at the 16S and we're wanting to focus on the V4 region. You could pick a primer set. So these green regions that are here represent the conserved regions. In the middle is your variable region of interest. You can design primers that will amplify from these conserved regions a wide diversity or a very large range of taxa that are present in your samples, amplify these variable regions, and then perform an analysis that actually can distinguish between the different taxa that are present, and then you can quantify it. So we'll take our toy example here. So we have a toy example of 4 microbes that are present in this sample. We extract DNA, and then you amplify it, and you get this stack of reads. Of course, in this case, we have it colored so you can see what everything looks like. You know that there are 4 different taxa, different abundances. And we have our set of reads, about 16 or so, that are present in the samples. Some of these reads will have sequencing errors, but many of them should be completely fine. They'll have full-length amplifications. You do also generate other artifacts in the data. I don't talk about chimeric sequences, but this is a real issue with PCR amplification. But you do want to try to address some of these when you're processing the data. So you have to somehow consolidate information, remove any artifactual sequences, or at least correct for them, and then try to get abundance information. So the general way that we actually do this for a targeted analysis is you just perform data reduction of some sort. You have to go through and essentially group the sequences based off of a percent identity, or try to at least generate a matrix of information which tells you that these sequences are alike and then correct for the errors. So you have the 2 methods. You have the clustering method, or the so-called OTU method, which has been popularized in tools like Mother and Chime, the Chime version 1, I should say. And then the denoising methods, which are much more recently in vogue and are popularized by things like Dada2 and Chime 2. These methods are going to compare the sequences to one another and generate basically clusters of sequences. And this is kind of where you start to see some of the influence of machine learning in the procedures. You're going to try to make inference from the similarities that sequences have to one another and then group them in some manner. So we'll start with OTU picking. The purpose is to actually cluster sequences based off of the identity. This 97% threshold is picked normally because that is able to distinguish organisms at the species level. Then you pick a representative sequence for the next steps. So you actually will pull out from that one group a single sequence. This is going to be representing what's called the OTU, or the operational taxonomic unit. The sequence is representing a group of closely related organisms. So it doesn't mean that it's the only representative. It means it represents this group. It's just something to keep in mind. The idea behind this is that you could have, our toy example has 16 sequences. And so in a regular example, you're probably going to have tens of thousands of sequences, if not more. And so you want to reduce the data complexity down considerably per sample and reduce the runtime. So you really want to make sure that you can actually analyze the data within the heat death of the universe is probably the best way to put it. So you want to have applications or methods that actually can increase the speed of the analysis, because you may have many different samples, but you also want to reduce the information that's present. And you also want to, this is a big issue, overcome sequencing errors. So in a TOR example, well, I'll show you a couple of examples of this. I'm not going to talk about the three approaches. The three approaches are of importance if you go down this path, but it's just good to know that in some of these you can actually include a reference database, a lot such as RDP or something along those lines, to help inform the sequences and assess whether or not you want to include novel sequences, depending on your analysis. There you go. So just to get an idea of how things are used, you have a number of different clustering methods. Depends on the tool. So Mother and Esprit use what's called a hierarchical clustering method. In fact, this is actually somewhat of a bottleneck because Mother in particular is known to be fairly slow. It does an all versus all comparison. If you have lots and lots of sequences, this can be an issue. The Mother was initially designed for use with 454 data, where you only generated maybe a couple thousand sequences per sample. With the advent of Illumina sequencing, now you're into tens of thousands, maybe even hundreds of thousands of sequences, depending on how diverse your samples are. And so it doesn't scale as well. So newer tools and methods have been developed to address this. Some of these use greedy heuristic clustering to speed up the process. A tool called Swarm uses agglomerative clustering, and Crop uses unsupervised Bayesian clustering. All of these are methods basically to make sure that you can cluster sequences together and try to infer and simplify data. So you start with this bag of sequences, including those that have sequencing errors present. You do a pairwise comparison. This could be using Mother, it could be using vSearch, uSearch, and so forth. And you generate basically a distance matrix and run a comparison against one another. You can cluster the sequences at 97% identity. So you'll get these groups. You'll notice that this cluster, for instance, is grouping things at 97%, but it actually includes your green sequence up here at the top, which I'll get to in a second. You pick a representative sequence. In this case, it's a centroid. So you try to find the center of the cluster and then find that particular group of sequences that's the closest to it, and you pick that representative. And then you essentially agglomerate or tabulate all that information, and you create OTUs. This has 3 OTUs. And in one of those, you'll notice that the red and the green is grouped together. That's because this may be a real difference, a taxon that's present that's different than the red, but the difference in identity is probably above that 97% threshold. It's probably a strain difference. And so OTU methods, you'll lose a bit of resolution. And that's actually an issue because you may lose some information at that level, and you may lose information that's valuable, such as if you want to distinguish between pathogens or non-pathogenic strains, so E. coli and Shigella, which are very close at the 16S level. If you happen to amplify the wrong region, that could be a problem. There's another issue, and that goes back to the picking of the sequences. So if you cluster the sequence, and you're doing the clustering based off of a set of samples, you're picking a representative sequence and then calling that your OTU. If you want to add newer data, let's say you're doing a long-term longitudinal study, you may have to go through and rerun everything and relabel your OTUs, because each one of those runs will have a different possible representative sequence. So it becomes a longer-term issue, and that's actually one of the reasons that led to this new application, basically a new pipeline referred to as denoising. So some of these denoising tools have been around for a while. the basic idea with denoising is that you're going to identify and deal with potential sequencing errors in amplicon data. So you're going to correct or remove errors that are present in those data sets, and then condense all the data down into unique sequences. So actually, instead of grouping things together based off of a percent identity, you're going to combine things into unique sequences. In this case, they're called amplicon sequence variants, or ASVs. There's a few other terms that have been floating out there. I think one was enhanced OTUs or EOTUs. But in general, I think the term that's used by the QIIME2 developers and the Dada2 developers is ASV. But in essence, what you're doing is you're combining the sequences down so that the sequence itself is essentially the identifier. So single nucleotide differences, the really common tools that are used in this case, Dada2 is probably the most well-known of these tools right now. It's in use in the new QIIME2 toolkit, the version 2 of QIIME. DBLUR is also implemented. That's a second denoising pipeline. And then the USearch tool set has also implemented a denoising algorithm called UNoise. Most of these are actually based off of the original tool, amplicon noise. This is a tool that was developed originally for 454 data, but is because 454 is really no longer in use, you don't see this tool developed anymore. But this is kind of the predecessor for the work that's been done on these. So how does it work? So essentially, this is it. So basically, you have the same kind of pairwise comparison. So you're taking that same set of data that we started with, and you're doing a pairwise comparison. The way that Dada2 does this is it uses the Esprit algorithm. So it's actually doing a hierarchical clustering, but it's a sped up version of it, looking for sequences that are very closely related. So you get the same sort of diagram. You basically get the same sort of clustering, but what you focus on is you focus on partitioning the data, starting with looking at the group of sequences that's the largest. That would be this red group of 4 sequences here. So you get this cluster right there. And the idea is that you're going to focus on that group because the probability of that group having a sequence error in it is extremely low. You can imagine that if you had a sequencing error present in that group of sequences, it has to be present for every single sequence. And so if it's a true sequence error, the probability is actually quite low, as opposed to, let's say, this sequence, where it's only present at a low abundance, and you can see that it's got one base change between them. So you start with this group, you compare all of the other samples, including these 2 sequences right there, to that one group, and you're saying, okay, what's the transition state that you have going from that one sequence, this small one, for instance, to the main group? And you're actually trying to see how many times you would have to change to get a sequence to be called a sequencing error. What's the probability that it's a sequence error? So this is an iterative process. You're learning the errors as you're going through, and then you iterate this over and over again. You basically do a partition iteration and learn the errors over a period of time using a particular threshold that you have to cross with the probability scores before you can assign, let's say, these 2 sequences to this group, this one sequence to this group, and it retains this information within these 2 groups as a separate ASV. So it's able to try to distinguish within a range of probability that a sequence that's present is either an erroneous sequence or a true sequence. When it groups everything, you get a same abundance profile, but you get higher resolution. So you get 1, 2, 3, 4, right? And then you get a higher level of resolution, and you generate an abundance profile. All of this information then goes into downstream analysis. So the advantage of this is that you're able to retain a higher level of resolution, But because you're looking at information at the sequencer level, you're not looking at information by just clustering, you can technically use the same procedure on many different independent runs and then essentially combine all that information together and do a larger run, because the ASVs, the actual ASV identifier, is actually the sequence itself. So they're much easier to scale up over time. And if we have time towards the end, I can try to explain it in maybe a little bit more detail, but we'll move on. All right, so taxonomic assignment. So we need to now take all of those sequences, either the ASV or OTU. We have a sequence, a representative sequence, or the ASV sequence itself, and we need to assign a taxonomic rank. So we got to say it's this genus or this species based off of a known database, such as Silva or the RDP database and so forth. I also want to point out Unite, which is a fungal database, which is another one that we've used recently. So we need to do a taxonomic assignment of information, and then we take these sequences to generate a phylogenetic tree. I'm going to focus on taxonomic assignment. I'm not going to talk about generating the phylogenetic tree because the assignment step is actually heavily involves using machine learning approaches. Specifically, it uses a naive Bayesian classifier called the RDP classifier. This was developed by the developers from the RDP, the Ribosomal Database Project, back in 2007. And the essential idea is that they take the known information from the Ribosomal Database Project, these known 16S sequences, and they've found that if you take the 16S data and you're able to take all the sequences and break them up into 8 k-mers of length of 8, you can actually use those sequences using a naive Bayesian classifier approach to classify sequences at the genus, up to the genus level, and you can do it with pretty decent reliability. So the way that this works is you break the sequence database here up into 8-mers. You find those 8-mers that can classify to the genus level, and then you basically take your ASVs. We'll go back. Here we go. We'll go to break your sequence data up into 8-mers, your input sequence, your ASVs or your OTUs. You have all of these strings of 8-mers, and you basically do a bootstrap confidence. You subsample those 8-mers that you generate, and you say, okay, out of these 8-mers, what is the confidence that I can actually predict something at the genus level? You do this 100 times, and you basically tabulate up the ranks. So you say, well, out of 100 times, how many times did it pick the genus? How many times did it pick the family? How many times did it pick the order, and so forth? And this is from the original 2007 paper. You can see that this is the length of the sequence here. This is the rank. And you can see that the longer the sequence, the more reliable of the assignment, which makes sense, of course, because you're getting more information. The V4 region falls kind of in this area. So you can get pretty reliable assignment at the genus level using the RDP classifier. Now, this is from 2007. And the databases have actually expanded quite dramatically. 2007 was right when Illumina sequences started taking off. And so I think that the database is easily increased by about 10-fold, if not more, even with just a focus on the full length 16S. So the methods need to be updated to deal with some of this. And what's happened over the last year is that the QIIME 2 developers have actually come up with a method that both re-implements some of the aspects of the RDP classifier, but also enhances it. So they have this new feature classifier that wraps the scikit-learn Python toolkit for machine learning. So what does it do? It focuses on, again, Bayesian classification, naive Bayesian classification. It uses multinomial Bayesian classification, so you can actually add weights to the model. The paper initially focused on this classification scheme. Because it wraps scikit-learn, you can also utilize other approaches. It has a flexible k-mer size, so you're not just stuck with 8-mers. You can change that. And then if you actually know what the frequency of the taxonomy is present in the samples, you can enhance the performance dramatically. As an example, this is a violin plot. I have to put a violin plot in here somewhere. So this naive Bayesian approach is the Q2 feature classifier. This is the RDP classifier. And you can see that these two are pretty similar to one another here. If you add in an expected taxonomic frequency, this is what happens with the new Q2 classifier. So if you're working in an environment or with a set of samples where you have an idea of what the abundance is or what the taxa that are present are, this works dramatically better than the other classifiers do. Of course, that's kind of expected, but if you're not interested in novel organisms, it's a huge improvement. All right, so at the end we have a table of counts. We have a table of OTUs and ASVs with their ranks. We have a table of experimental data, and we have a phylogenetic tree. This goes into data analysis. So I'm going to transition over now to shotgun metagenome analysis. And we'll probably focus on that for a good chunk of the rest of the time here. So you're going to see that with the targeted analysis, you condense the data down. You get counts, you have taxa, you have metadata, you have a phylogenetic tree. You're trying to get to a similar step with shotgun metagenome data. You need to get to a point where you can quantify information in some way. This approach is, of course, sequence everything. You're really just pulling down and sequencing as much information as possible. The advantage is that you get both taxonomic and functional classification. So you're going to be able to actually look at things at the gene level. There are two broad approaches for this. You have a direct read analysis approach, and then you have an assembly-based approach. Looks like some people dropped out there for a second. Is everybody okay? Everybody hear me okay? All right, cool. I just saw a bunch of people drop off the participant list and then come back for a second. All right, awesome. All right, so you have two different approaches. You have a direct read analysis approach, and then you have an assembly-based approach. I'm going to focus initially on assembly because it's actually the most complex, but we'll start with our toy example. Again, here's our toy metagenome with 4 taxa. You're extracting the DNA. You're going to generate a ton of sequence data. So in comparison with the targeted approach, per sample, you're going to probably have more like 25 million reads. So it's way, way higher. You're trying to capture all the genic content. If you have a very diverse sample, it may be even higher than that. And then these can go into the assembly approach or the direct analysis approach. So with assembly, you're going to start with the whole metagenome sequence data. You're going to assemble this into scaffolds. You're going to use a genome assembler that's specifically developed for metagenome assembly. You may have more than one sample. If that's the case, you're going to do what's called a co-assembly, where you're going to try to combine all the read information or all the assemblies into one large assembly. The main reason to do that is so that you can do comparisons. If you have different assemblies and you want to do a comparative analysis, it becomes extremely difficult to combine them. So, and there's lots of methods to do it, but they're not consistent. So it's best to do a co-assembly if possible. So you get this big bag of scaffolds. You want to then take your original raw read data, and then you're going to align it to this. So you're going to get BAM files along with your assembly. This generally goes into the binning step, which uses a machine learning approach to combine information. And then you're going to go into annotation, where you're going to use machine learning approaches to annotate the data. So we're going to focus initially on binning. So with binning, the idea is that you want to somehow combine information in a manner that you can detect novelty. And with that in mind, you want to use an approach that is not dependent on taxonomy, at least initially. You want to use an approach that uses information derived primarily from the sequence data itself. Because if you use something that's taxonomic dependent, you may actually miss information that's not present in the database. In other words, novel sequences. So the general idea is that you can use abundance information from the alignment step. So you have coverage that you can get from the alignments. You can use feature information from the scaffold, so GC content, tetranucleotide frequency, and then combine that or use it independently depending on the tool. So you can use a number of different methods for actually clustering the sequences together. And you use this as basically as input. You can see feature extraction here. So that information can be used in a machine learning model to essentially cluster the data together. And it uses, each of these tools uses a wide variety of different methods. So most common one I believe is k-means, but the general idea is you're grouping things together without including any kind of taxonomic inference. Now, this is a general grouping. If you had something at the strain level, for instance, you could imagine that GC content will probably be very similar. It's feasible that the abundance might be similar enough, and so these could be combined together into the same bin. So you can use a second tool that uses hidden Markov models called CheckM. CheckM assesses the quality of metagenomes, and it uses single copy genes within phylogenetically distinct lineages to assess the quality. So what CheckM essentially does is it looks for, if you have a cohort of hidden Markov models, and that represents a single copy set of genes within a particular phylogenetic group, you can have it scan the sample of interest and assess whether that bin has all of those hidden Markov models, so it's complete. But if it has more than one hit for that hidden Markov model, that may indicate, because it's a single copy gene, that you may have more than one genome sequence or organism taxon that's present in that sample, in that bin. So you get this idea of completeness and contamination within your sets. And you can do this actually with regular genome sequences, by the way. And so I've seen people use CheckM to assess the completeness of a standard genome sequence. It's developed primarily for bacterial sequences, though. So once you have that information, you can decide how you want to actually deal with it. So do you want to have a bin? This is an example from this paper where they assessed that this one particular bin was heavily contaminated. You can see many little clusters in here. And whether or not you want to take that bin and clean it up so you can split it up and try to pull apart those individual clusters within that bin, or whether you want to proceed forward. They actually came up with an iterative pathway to try to sub-bin this data. So you can imagine with the metagenome assembly, it gets extremely complicated, because this actually, if you get a very diverse sample, you could go through this whole process of just resolving bins. Once you've resolved them, which hopefully will happen before the heat death of the universe, you go into annotation. Annotation uses standard gene calling tools. These use hidden Markov models or neural networks, but there's a modification that's included with these gene callers. So Prodigal, GenMark are tools that have been used for bacterial or identifying bacterial genes. But in most cases, these use or look for an open reading frame. And then with metagenome data, it's highly possible, highly probable that you'll get more fragmented scaffolds. And so you need to identify sequences that could produce a protein sequence even if there is no open reading frame. So you have to modify the models. So you have metaprodigal, metagenemark. FragGene scan is also a very commonly used one that was developed specifically for metagenome data analysis. Once you have the information, you have a protein sequence, you can then use information from databases to infer taxonomic and functional information. And so you can start actually assigning these different taxa. And this is a very scaled down version of the KEGG pathway map, but but you can start inferring functional information as well. Direct read analysis is actually one of the only ways that you can assess if you've got hundreds of samples of metagenome data. Because doing full-blown co-assemblies on hundreds of samples with billions of reads is not recommended at this time, although some labs are actually trying to produce procedures to do so. But the direct method is essentially just taking the shotgun data, so we go back to the original raw data, and you just literally assign taxonomic and functional information by combining or aligning these to a particular database. If you're only interested in taxonomy, you can use things like centrifuge, cracking, kaiju, metaflan, or diamond to align these. These are not machine learning based tools. They're basically looking at, they're doing an alignment approach and just looking for information in a known database of interest. However, there are some tools. NBC is a naive Bayesian classifier, but developed as opposed to the RDP classifier developed for whole metagenome data. Thimble uses a hidden Markov model approach. TIP-SEP and HIP all use hidden Markov models. TIP-SEP and HIP, by the way, are developed by Tandy Bohr now here at UIUC. And all of these tools are using machine learning approaches to add information. I will say that these approaches tend to be slower. And so that's one of the reasons that you don't have that. And I should mention, Human 2 is one that was just recently published. It uses a database called Chocoflan. I couldn't find a logo for it, so I just stuck a picture of the Chocoflan dessert from the Betty Crocker website. At the end, what do we have? You're actually not that different if you use a targeted approach versus a whole genome approach. You're going to end up with one advantage. You'll have an annotated metagenome if you do an assembly. It'll be split into bins, but you will still have some kind of matrix with IDs and counts. These IDs could be genes, it could be taxa, it could be functional. And then you're going to have associated functional data, a table of experimental data. All of these can go downstream into statistical analysis. So beyond the metagenome, you can do metatranscriptome, metabolome, proteome. There's a lot of additional information that you can add. There's a lot of additional things that you can perform, and you may want to think about if you are trying to establish whether a particular microbiome is influencing the environment. So you may want to look at metabolites that are produced as an example. And this is very commonly done now. I've seen a number of projects personally here where they have been trying to integrate metabolome and metagenome data, or looking at expression with metatranscriptome. So next is data analysis. You basically get count information. You can do simple ordination plots to try to actually see if you have an overall pattern. You can get composition to see if there's a change in the overall composition based off these experimental groups. This is actually a whole metagenome analysis here that we recently did, and it's looking at changes in experimental groups. Look at alpha diversity, beta diversity, which involves ordination. One of the most commonly used methods downstream is random forest. And that is so you can actually use and train data sets to actually predict a particular phenotype. But one of the neat things about random forest is you can also use it to try to identify particular features that influence the model. And this is one way you can hone in on specific groups that may play a role in how you go from a, let's say, a disease or a control state or a normal state to a disease state. This has actually been used fairly extensively for prediction of disease. So there's one example from a few years ago, a number of years ago, from actually the BGI group, where they used the random forest model to predict whether or not a particular set of samples was probably a carcinoma. So they had control samples, advanced adenoma samples, and just based off of the microbiome content, they were able to actually make a prediction that that these are likely going to be carcinomas. This has evolved to the point where Nicholas Agata's group developed a toolkit called MetaML, meta machine learning, basically focusing on phenotype prediction. And so they've sort of elaborated on that previous study even more so to actually point out that you can use random forest models to predict with pretty significant degree of confidence whether or not a sample is coming from a normal versus a disease state. And so this is a standard ROC curve. You can see in some of these cases, the cirrhosis and colorectal cancer samples, the true positive rate is actually fairly good. This is an SVM support vector machine approach in comparison. And they found in this study that the random forest model did a really awesome job in comparison with the SVM. Some of these work better. Cirrhosis, actually, you can see across the board, did well. Some of these aren't as good. Obesity wasn't as predictive, but you get the general idea. So I'm going to go through the challenges very quickly. The challenges that I see we will be facing are larger metagenome projects, evolving DNA technology, integration of multi-omic data, and then just keeping up. This is the issue that we face. That's a MiSeq nano run. So that little open spot there is 1 million sequences. So if you were to do a 16S targeted analysis on 40 samples, that's probably more than enough. If you have a high-seq 4000, you got 8 lanes. Each one of those lanes is 400 million reads. This is the NovaSeq. And that one lane I'm pointing to there generates 6 billion reads. That one lane generates that's one lane. So obviously you can get 24 billion reads off of a single run. These get processed in I think a day and a half. So you have 24 billion reads that can be generated in a day and a half. It's an incredible amount of data. And it's actually much cheaper to sequence this way. So there are projects that are actively looking at using these data sets so they can increase the number of samples, and this increases complexity of analysis. Evolving technologies, PacBio, that's an Oxford nanopore, probably at scale. So it gives you an idea of the differences. But long-read technology is heavily influencing what kind of molecules you want to look at, such as full-length 16S. There is a protocol called Loop that is they're looking at methods to make linked reads for 16S. They have ways to generate multiple amplicons in one run. So you could have one sample in a split-arm axis array that can actually have up to 48 different amplicons. So you can do many different regions of the 16S, you can do ITS, you can do up to 48 different things for each sample. Integrating that data is a challenge. And then high-C and 10x reads can actually be used to generate much more reliable metagenome assemblies by combining information that links information together more reliably. A lot of these technologies are extremely expensive, and so that's an issue. And very quickly, multi-omic analysis. This is an area I think that is going to very likely benefit from things like deep learning. What are the best methods for combining all this information? There are publications that are really starting to come out on this. This is one that just came out in January. Actually, I think Steve Quake and Michael Snyder were involved in this, and they used elastic net approaches to analyze the data and look at changes during human pregnancy, during the gestational period of human pregnancy. It's a really interesting approach. Deep learning is on the horizon, I think, for a lot of these data sets. And then finally, just keeping up. This is how I feel like on a day-to-day basis, because it really is a challenge to try to keep up with this. But it's a really cool area of research right now to be involved in. It's exciting. But yeah, just trying to keep up. It's a full-time job in and of itself. So with that in mind, I'm going to wrap up. This is the Biotech Center. Biotech Center is actually my home base at the university. I want to thank Jenny, Jessica, Gloria, Kim, and Chung, Luda, Prakruthi, and H3A Bio. And with that, any questions?
Thank you so much, Chris.
Thanks.
You didn't disappoint. I've been seeing on Twitter and other social media pages that people have been looking forward to this talk, and you didn't disappoint. So thank you so much. We're open for questions now. We have about 7 minutes remaining where we can engage with Chris. I mean, as you can see, you can follow him on Twitter @CJFields, or you can email him, and he'll gladly engage with you. But for now, based on the talk that we just had, and I understand that it's obviously some of you might need some time to mull over certain things, but for now, if you have a question, please raise your hand. At the top of your screen, there should be a person raising their hand or an icon of a person raising their hand. Click on that. Alternatively, you can type in the chat room if you have any questions, and Chris will be able to answer your questions. No, you didn't. I mean, I don't think you
scare everybody.
It was so interesting, and there was obviously a lot of information. Someone is typing. Let's see, James is typing. James, you can alternatively raise your hand. But yes, you can type the questions and I'll read them out for Chris, and he'll answer them, sorry. But yes, Chris, I don't think you scared anyone. You just obviously gave us a lot to chew on. Shakun says, thank you, Chris. A very clear and interesting presentation. Thank you so much, Shakun, for that comment. Are there any questions? I'm going to give us about 3 minutes, and if there aren't any, obviously not going to take them out of you. Oh, James, people are typing. Sorry, let me be patient. Let's wait for people that are typing and see what they're saying, and then I'll see what we do from there.
Are you talking about with it depends on where you're talking about the clustering step. Are you talking about at more deriving OTUs or ASVs, James? I'm guessing that.
Busy typing to clarify what he meant. OTU. He meant a.
Yeah, I could go back to that slide real quick because it's actually pretty simple here. Actually works the same for ASVs as well, but essentially, so you have a set of sequences here that are actually quantitatively, so actually is something I probably should have mentioned. The circles here are actually relative to the quantity of the sequence that's present in this location. So the group that's in this cluster, you can actually quantify by just tabulating up the number of times that sequence is represented in this group. And so you pick that, all the sequences that are in this cluster, and then combine them to get an abundance profile. But what you do is you just have this one specific representative sequence that represents that. Does that make sense?
James, I hope that makes sense. And Charles has a question. Chris, Charles is saying, how can we apply machine learning in RNA-seq to predict patient survival?
Oh, so well, I think it's it's probably not a dissimilar approach, though. I don't see we typically don't see or haven't seen a lot of use of machine learning to do this yet. But you could probably use a similar approach like random forest to assess whether or not a particular set of features, such as a set of genes that are important in going from or being able to classify samples in a control versus a disease state. Does that make sense?
Alfred says, thanks a lot for the talk, Chris. Any comments about dealing with contaminants and microbiome data? It's a real big issue around this topic. Do you have anything to say, Chris?
So you talk about microbiome like in background samples, like in water samples? Or are you discussing about things that get misamplified or get pulled up? Because if you're talking about contaminants in water samples, there are actually tools that are developed to deal with background amplification that you get from water. If you have contaminants that are being misamplified, there are some methods to actually deal with looking at, let's say, sequences that don't actually align very well to the ribosomal RNA database. There are methods to assess, but it comes down to deciding what's the proper bootstrap confidence value that you want to use. But if you're looking at contaminants in microbial samples, there's a R package called decontam that can actually remove background that we use.
Okay, thank you, Chris. I'm gonna allow for 3 more questions. I'm gonna read 2 more that I see now, and if there's anyone else, you can ask, but in the interest of time, I'm gonna ask that we follow Chris on Twitter and that we email him. And just to, again, say, this is being recorded and we're gonna make it available on YouTube. So it will be available for you to also see at the end, like if you wanna, in your own spare time, or if you wanna share with other colleagues and networks, you'll be able to do so. For now, Gilbert is asking Chris, he says he's working with insect CO1 barcodes, roughly about 200K sequences. He's asking if you have any tools that you could recommend that.
Yeah, I mean, you could use you can actually use Data 2 for non-16S data. We've used it for custom primer sets, so technically you could use it for anything. It's supposed to be a general denoising tool that can be used for any amplicon data. So I would recommend trying it. It depends on whether or not the amplified sequences that you're using are, well, they have to be Illumina. That's what it's designed for if you're using Data 2. And the reads should overlap. You should have one potential contiguous sequence that comes from the sequence data. Charles? Yeah, I was going to say, Charles has got the question about machine learning platforms. Scikit-learn, I would say.
Okay, sure. Okay, cool. Thank you for that. Jonathan is typing, so I think we'll give Jonathan a few moments to finish typing his question or comment, and then I think we can wrap up. Okay. Jonathan, we're waiting for you.
Machine learning based patient prediction tools. Yep. What are the underlying issues for outcomes? I mean, I think you see the same problem with microbiome tools. I think that this is an ongoing area of research. This is that MetaML, and you can see in some cases machine learning approaches work better with some samples than others. And depending on the approach, some of them actually work better than one approach versus another. So I think this is going to be an ongoing area of research. I think that more complex models are probably required, especially when you have something as complex as a disease state. So random forest and SVM I think work fine, but there are probably better ways to get at data, and this may be an area where deep learning I think is going to play a significant role, using more complex machine learning approaches, I should say. Does that answer your question?
Okay, Chris, thank you so much once more for such an interesting and informative talk. And I'm sure you're going to get more followers on Twitter now, and your email box is going to be filled with queries about this. I just want to also thank everybody who attended in different countries, in different classrooms in their numbers. Really appreciate you joining our monthly webinars. We're going to have another one in April. We'll just inform you either through social media or an email about who's going to be the speaker. But for now, thank you so much for attending and enjoy the rest of your day. Goodbye, everybody.